That should be fine.
Okay. Okay. So I'm actually being fascinated about what we can publicly see about your journey into antiviral gene therapy. But a place I would like you to start from is your entry point. When did you start becoming really, really curious about this line of research?
Paul, after I finished my medical studies here at Wits, I did a PhD. I was more interested in basic science than in clinical medicine, and I studied hepatitis B virus and its relationship to liver cancer. After I finished my PhD, and that goes back about 30 years actually, I was looking around for opportunities to broaden my horizons as far as work that I was doing is concerned. And it was recommended to me by my PhD supervisor and other people that I go and work in a laboratory in Paris, in France, in a hospital there called Necker Hospital, which is the main children's hospital in Paris, in the laboratory of a man called Christian Brescio. Um, and I went there, and when I arrived, I didn't know what to expect. He was very keen to set up gene therapy as a new discipline within his quite big research laboratory. And that was really when I got involved, and I worked with another researcher there called Nicola Ferry, who was the more hands-on, experienced person in gene therapy. And we did some work on trying to develop new ways of treating liver cancer using gene therapy, engineering sequences, engineering DNA to make genes that would treat or kill cancer cells specifically. And then when my postdoctoral fellowship ended, I returned to South Africa, and I was really very interested in gene therapy. That sparked my interest, that experience as a postdoctoral fellow. And when I came back to South Africa, I set up this laboratory, which is now the Antiviral Gene Therapy Research Laboratory. And we have been focusing on viruses, mostly hepatitis B virus, not really on liver cancer, which was the field of my postdoc fellowship. And we've done work for many years now on trying to develop new ways of disabling viruses, hepatitis B, as I mentioned, hepatitis C virus, HIV, Rift Valley fever, and one or two others as well. So we've been using methods that can be described as gene silencing or gene editing to disable the genes of different viruses, and we've had some success with that, and it's gone quite well. Um, but what is interesting recently is that after the pandemic or during the pandemic, as you know, mRNA vaccines became very popular, and it was shown that they're very effective. And we had some experience with making mRNA as part of our gene therapy research. So our laboratory has now sort of shifted its focus from therapy to vaccination. Yeah, so we're very involved with developing new kinds of vaccines based on using mRNA particularly, and also engineering viruses, adenoviruses, such as the viruses that were used by AstraZeneca and Johnson & Johnson to prevent infection with SARS-CoV-2. So that's, in a nutshell, the background to where we are now in the laboratory.
Okay, that's a very, very interesting and probably succinct summary. In March, you and several others published a piece, correspondence, I think, in Nature Biotechnology,
Oh,
in
yes.
which you were talking about making genome editing a success story in Africa. And I think when we talk about genome editing and Africa, for people that are not really uh, proponents or really realizing its potentials or the reality of its adaptation in Africa would think it's still several years away from being realistic on the continent. But we also see a good case in point is the recent the development for sickle cell treatment,
Mm.
genome editing. When considering the fact that Africa has a higher population of individuals living with this disease than elsewhere, that this innovation can actually be accessible now begs the question of the fact that Africa should probably not be seeing this innovation to be lots of years away. So where do we stand currently? And for somebody that thinks, what is your position on letting this work on the continent, and how does that journey look like from your perspective?
My own feeling is that it's very important that African researchers, African scientists get fully immersed in this new technology. It's gone on for far too long that Africans have waited for people in Europe or North America or other parts of the world to develop therapies, such as you quote, for sickle cell disease, and then take what other people have invented to apply to the problems in Africa. I think that is inappropriate. It's African science needs to modernize and needs to address the problems that the continent faces, and its own scientists need to be immersed in developing solutions. And I think sickle cell is one example, but the other one, getting back to the pandemic, it became clear at the start of the pandemic that Africa was completely inadequately prepared for the pandemic, and as a result of that, vaccines were not available quickly. Many people died as a result of that, which is unacceptable. And so that's really why the WHO had this project to make sure that African science, and in fact science in other low and middle income countries, is developed so that the health security is improved. So to answer your question, I think that it's vitally important that African scientists address problems, obviously in collaboration with more experienced laboratories in North America and Europe, but it needs to take the reins and be in charge of its own solutions.
Okay, then I'm curious to know about how you see the decision of what approach to prioritize in responding to diseases. We have, for loads of years, we've seen immunotherapy actively advancing in cancer, for instance, and also transitioning in some aspects to HIV. We see how the HIV to vaccine ecosystem continues to strive to tap into the immune system to activate as much immune response as a preventive measure for HIV. We've seen loads of advancements in this over and over again, but I would argue that we've not comparatively seen consideration for gene therapy, antiviral gene therapy, as a way to answer some of these protracted questions. Now, looking at the different approaches that you are talking about, the different applications, other routes of addressing these issues, they seem to have really accelerated over the years, and the ecosystem might have gotten so used to considering them as the only way to find answers to the questions that we are still looking at in terms of developing new tools. What is the argument in favor of antiviral gene therapy as a way to still move forward?
Look, Paul, the resources of gene therapy for disabling viruses have increased considerably over the past few years. So there's a big toolbox, if you can call it that, of things that people can use to inactivate viruses. And it's becoming easier and easier to do, although it's still quite expensive. It is becoming more tangible, the use of gene therapy to disable viruses such as HIV, as you said, and others as well, HBV, which has been our particular focus. So the technology is advancing quite quickly, and it's becoming more accessible to LMICs, such as the countries in Africa. And I think it's a case of sort of similar to RT, where the cost of computers, the power of the computers has increased considerably over many years, but the cost has basically come down as well or stayed the same. So I think that it's going to be probably something similar with gene therapy. At the moment, it's very costly and quite complicated, but as the field matures, it's going to become more accessible. At the moment, sure, it's a sort of frontier type technology, but I think it's going to enter the mainstream very soon as the technology gets easier to apply.
Okay, I'll take that. Then let's talk about the progress that has been made in HPV at the moment. So, what was the landscape like when you started? Where is it right now? And what is promising in this line of research? If you can even localize it for the African continent, that would be awesome.
Yeah. So the article that you referred to before, that I was the commentary piece that I was a co-author on, we cited work that we'd done on using gene editing. That was the topic of the commentary piece, to change the genes of hepatitis B virus so that it could no longer replicate and thereby cause a cure. Hepatitis B virus is, like HIV, a very difficult virus to treat. And there's some similarities between HPV and HIV in that the DNA that's in the cell, in the liver cell, that is responsible for expressing all the genes that make the viral particle, is very stable in hepatitis B virus. It's not quite the same as what's in HIV. HIV has the so-called provirus that integrates into the host genome, and that's what is used to make the viral particles that perpetuate the infection. With hepatitis B, it's a piece of DNA that's in a circular structure that sits in the nucleus of the liver cell, and it churns out the virus constantly. So you can treat the patient with antiretrovirals, just like with HIV, reverse transcriptase inhibitors, and you can suppress the replication of hepatitis B virus. But as soon as you withdraw those drugs, then there's rebound and the virus starts to proliferate again. So a more lasting solution is to attack that DNA that is responsible for making these virus particles and to change it, to mutate it so that it's no longer active. And that was the work that we cited in that particular paper. We've been doing quite a bit of it on using gene editing to mutate genes of hepatitis B virus. And there has been some success with that. Certainly with the preclinical work, we show very, very good efficacy, and we want to take this forward to evaluation in a clinical setting. There are a few questions that need to be answered related to this, and that is that we need to make sure that it's very specific for the hepatitis B virus gene, because we don't want the gene editing to change something in the human genes, which could be disastrous, obviously. So that's quite a big question that needs to be answered, and that's really what the focus is at the moment of our work. But certainly we've shown through proof of principle that this is feasible. Certainly in preclinical studies, the virus gets basically completely disabled and it can't proliferate anymore, which is quite a significant achievement, I believe. Of course, there are other kinds of treatment that other people are working on, all sorts of different targets that they are working on. But our field in Africa, obviously, the gene editing approach is showing some success.
Okay, then how do you see this research helping the people? I remember when the case argument for gene editing in malaria is to edit the genes of the mosquitoes, make it unable to transmit malaria, and gradually release these into the market. We could see, we can remember the lots of pushbacks that's had and continues to have. And do you think we've gotten over that amount of pushback for mosquitoes to now seeing potential acceptance of crossing that to the human genome? Or how do you see this playing out? Are we going to be targeting the virus genome to edit it? Or are we going to still, do you consider editing the genes responsible for the host response to these viruses as a better approach?
You raise a very good question. it's crucially important that the technology be analyzed thoroughly to make sure that it's absolutely safe, so that people don't have to be concerned about their own safety if they receive this kind of treatment. And I think that it's probably, in the case of gene editing, it's probably not quite there yet, certainly for hepatitis B virus. Just to pick up on the last point that you made in your question, the gene editing approach that we're working on does not target human genes. It's only targeting the hepatitis B virus. And I think that that is probably the better approach to take, because it's less risky. You don't want to, I think it's not good to change the genes of humans to make them resistant to HPV infection or whatever else it might be. I think it's much better to target the genes of the virus itself. But, if this technology matures and is shown to be safe, which we certainly hope it will be, and the indications are that it will, then it's also crucially important that the public be informed and educated about this kind of treatment, and what the potential shortcomings are, where there might be symptoms or problems resulting from the treatment. So it's crucially important to have the full buy-in of communities to make sure that they understand the treatment and that they can accept it. Obviously, it's got to be safe. It can't do anything until it's proven to be safe, because Sub-Saharan Africa has got a huge number of people who are chronically infected with hepatitis B virus, and it's causing problems of cirrhosis and liver cancer, which are very, very serious complications that are usually fatal. So it's important that a new effective treatment gets developed, but at the same time, we need to be sure that communities throughout the affected areas are well informed and understand what the treatment's all about.
Yes, I'll take that. Then, before going elsewhere, still zeroing in on hepatitis, which space we are seeing African countries massively rolling out? There are vaccinations early stage for the child, vaccinations being considered as one of the ways to actually prevent this. We are seeing aggressive publicized awareness being made in this, and I think we're also seeing an handover, what will I say, associated improvement in public awareness around vaccinations for liver cancer, for hepatitis B vaccine, in similar way that we are looking at the prevention for other cancers. For instance, look at how fast the adoption for cervical cancer vaccine is also accelerating. Yes. So, looking at all of those progressions, how do you see the prognosis of this disease, and how do you see its potential eradication path, or eventually masterly good toolkit looking like?
Again, that's a very good point, Paul. Vaccination works very well. The vaccines that are available do prevent hepatitis B virus infection very effectively. And it's important that the children be vaccinated against hepatitis B, because if a child gets infected with hepatitis B, then the risk of becoming a chronically infected individual is very high. And it's the chronically infected individuals who get liver cancer and cirrhosis, which are the serious, often fatal complications. So it's important that the vaccination program be effective. The problem is that actually it's not that good, because there are problems with, say, the regimens that are used for vaccination timing. For example, in South Africa, the vaccination against hepatitis B virus started in 1995, which is a long time ago, almost 30 years ago. But it's only now that the vaccination at birth is being implemented. And that's quite an important time to give a baby a vaccination, not wait for 6 weeks when the baby may have been exposed to infection from the mother, and then the child would already be infected and then become a chronic carrier later. And bear in mind, of course, that the vaccination is not therapeutic; it's preventative. So it doesn't matter if you give vaccination to a person who's already infected, because they won't eliminate the virus. These vaccines that are licensed now do not eliminate the infection. So it's a problem. So as I say, the vaccines are pretty good, but I don't think the implementation of the vaccine programs has been particularly good throughout Africa. So the problem of new infections, chronic infections, is actually not getting resolved. So that's why it remains important to develop a good treatment, as well as to work alongside improved vaccination programs.
There's also this issue that I want your thoughts on, which is the perception of gene therapy and others that seem to be in the line of made tailor-specific innovations to not be very, very good in defining public health response. What am I trying to say? Look at what is happening in the way precision medicine is being marketed or is being raised awareness about, which is an intervention that is specific to what you are dealing with as a person. And like you've talked about antiviral gene therapy, we see a potentially adaptation to a region. For instance, there are uniqueness around the viral genotype. Okay, let's go. So let's do some. If you look at what we currently have with Mpox, we have clade 1, we have clade 2, we have clade 1b, and these often start from a part of the world before spreading to the other part of the world when this eventually happens. Bringing this into hepatitis, we could potentially see this, or do you think this particular evolution and emergence of new types of these hepatitis, clade or whatever uniqueness approach would actually put this particular approach of antiviral gene therapy development into jeopardy? Or do you think antiviral gene development therapy approach by design is tailored to actually address these potential problems?
Yeah. The hepatitis B virus isolates that are found in Africa are different to the isolates that come from other parts of the world, and in particular Asia, which is where the infection is common as well. So it is important to tailor the therapy to the features of the viral genotypes that are in a particular part of the world. And not only that, it's also important to tailor the therapy to the human host as well, to make sure that they do not react badly to this therapy. But just to answer your question about the evolution of these genotypes, and you talk about Mpox as well, one of the very good features of gene therapy is that it is highly flexible and it's based on rational design. So it's possible to take a therapy, test it. If it doesn't work, it's possible easily to change that therapy to make sure that the deficiencies of the first version of the therapy are addressed, and you can actually improve on it very quickly. And it, as I say, it's based on rational design, and you can adapt therapies to cope with problems of evolving viruses, new genotypes, whatever it might be, new variants of concern, and also to deal with specific features of certain populations. So, yeah, I think you highlight a very important point. But one of the advantages of gene therapy is that it is possible to adapt quickly and pivot and change some variables to make sure that the problems are mitigated.
Then do you consider antiviral gene therapy as a potential public health solution that checks most boxes of what public health solution should look like in terms of adaptability, in terms of being able to help as many members of the general public as possible?
I think that would be a wonderful thing. Obviously, the big uh, problem at the moment is the cost. It's really difficult to roll out gene therapy for public health problems right now because the cost is prohibitive. There's no doubt about that. But to get back to the RT analogy, I think the costs are probably going to plummet when the technology is better worked out and the new innovations that come that will lead to drugs being made that are cheaper and more affordable in Africa. So I think that's certainly something that I would love to be able to see, and I think it's a realistic goal to have. It would be wonderful if that was achieved.
Okay, I'm also interested in having this conversation around with you, which is looking, adapting innovations that have reached a lot of progress for other types of research into virus intervention. I think you started really well with the cancer analogy, looking at what is being used for cancer and now bringing it into antiviral intervention that is locally relevant on the continent. We've also seen this playing out even in immunotherapies. And we are seeing HIV, new approaches to HIV, taking a look at what seems to have been tested or deployed for cancers. What is your position? What are your thoughts around taking what has been tested for probably non-communicable diseases and bringing it into communicable diseases to potentially solve this particular problem? And this could actually be potentially bringing attention to what actually in African countries where government continue to be laser focused on communicable diseases as what to spend money on, as what to prioritize. Meanwhile, non-communicable diseases are serving as a tool development platform for communicable diseases. So what are your thoughts on this interesting perception?
Non-communicable diseases are undoubtedly very important, and people are starting to appreciate that and not focus exclusively on infectious diseases or communicable diseases. And I think it is important that the non-communicable diseases get the prominence that they deserve. And I think that non-communicable diseases are benefiting from developments in gene therapy. You talk about immunotherapy, and that's been a very good line of work to prevent or treat cancers of various sorts. And that's a kind of, these engineered CAR T cells, which is commonly the approach that's used to treat various kinds of cancers. That has its origins in gene therapy as well. So gene therapy has sort of spread its influence far and wide. It's not just in infectious diseases, but also non-communicable diseases. And I think as the technology improves, it's going to have a bigger influence. And I said before, it's going to reach the mainstream of modern medicine. And it's the principles of gene therapy. And as I said, its big, strong feature is that it's based on rational design. So you can design a therapy based on information, particularly about sequences, and implement it pretty quickly. And it's gaining application in non-communicable diseases, certainly.
Okay, then I'm also interested in convincing government or funders to commit resources and funding to your line of research, especially the fact that much comparatively lesser noise is being made in the media and those that ought to actually help you attract attention to it. I know you've spoken at a lot of conferences. You also work with the SAMRC. So what does it look like trying to secure funding, support, and bringing attention to your line of research?
That again is a good question. I know when I came back from my postdoc in France and started working on gene therapy here in South Africa, there was a lot of skepticism. Skepticism, not only from the lay public, but also from my colleagues. They sort of said, why are you doing this work? It's not relevant to Africa. Leave it to the North Americans or the Europeans to do this work. Um, but I think attitudes are changing now. Certainly, after the pandemic, when it became clear that knowledge about gene therapy has got application to preventing infections such as what was causing the COVID pandemic, SARS-CoV-2. So, knowledge about the gene therapy was very important to try and prevent this infection. So I think attitudes are changing, and people are also realizing that it's important that LMICs, African countries, need to have these resources at their fingertips to improve their health security. It's obviously a lot more rewarding if a country can solve its own problems, and it's also more practically beneficial. I think it's not good to rely on outsiders to give their inventions, or charge for their inventions to treat diseases of African importance. So I think attitudes are changing, and as I mentioned before, we have been involved in this initiative to develop mRNA for vaccines, and that was spearheaded by the WHO, World Health Organization. And they've pushed it very hard, and they've put in a lot of support and got a lot of governments, European governments, African governments, and I think Canada as well, to give support to this initiative. So I think funding is improving for this kind of work, and it's taken quite a lot of time, and also the experience of the pandemic and the influence of people who are very important globally, such as from the WHO.
So probably my second last question is, what changes do you still desire to have in this to make the journey to getting the best out of your line of research, make it much shorter and more promising than what it appears to now be at the moment? So what changes are you still looking forward to?
I think that relates to your earlier question as well. Funding is crucially important. We need to make sure that there is adequate support for scientists in Africa to develop the skills, the resources, the know-how to do this kind of work. I suppose it's not only gene therapy, but other kinds of sophisticated modern medical treatment development. Funding is crucially important. We need to get the support of governments, policymakers, who realize that it is essential that these skills be developed in countries, and obviously we need support from the so-called global north to assist and endorse this kind of work as well, to collaborate to facilitate the development of the expertise.
Then what about the future? Looking at the future crops of scientists in terms of masters, degree students, PhD. So what are you? Does a pipeline of talent look promising?
Yes, well, my career has basically been mostly involved with training PhD and MSc students, and the people coming through the pipeline are always very impressive, and I think that they still continue to impress me. And that's something that is crucially important. You need to keep the training up, make sure that the youngsters are getting the training that they need to be able to embrace this new technology, and that they can then develop into leaders. And it is a challenging, demanding thing, because it's not the most simple of fields, gene therapy, that is. It requires a lot of dedicated training to bring a scientist up to speed with how to do gene therapy. But it is crucially important, and I think that the human resources are developing. I think compared to what they were like, say, when I started this work back in South Africa in the mid 1990s, there's a huge difference. What the capacity is now is vastly superior to what it was initially when I started. And I think that it can only grow from there. Another thing which I guess you've gathered from the tone of what I'm saying is that this is a hugely rewarding and satisfying field to work in. So youngsters who start working on gene therapy and they develop something which they say found to work, say in a animal model of a disease, that's hugely satisfying, and that builds confidence, and it also spurs them on to be more ambitious, and that's a really good feature, I think, of gene therapy, which I think is playing out now. So the critical mass of people who can do gene therapy in Africa is improving all the time, and let's hope it stays on an upward trajectory.
Yeah, and this one has one here and one B, but let me put it together. I see that you still have some association connection with the cancer community in South Africa and probably other parts of the continent. What does that currently look like for you in terms of what you currently do with hepatitis and the focus of the cancer community? And probably the very last question is, what is Africa's unique positioning in this line of research with antiviral gene therapy? What answers do you think could probably only be gotten from Africa that the rest of the world can benefit from? That could be the basis for continual justification of this research and encouraging attention and resources to be committed to it.
Yeah, it's a truism that Africa has always got something new coming from it. I think it was Pliny the Elder who said that the Roman 2,000 years ago odd. And I think that the pathogens that, say, come from Africa are very important for the rest of the world. And it's important that the African scientists are aware of what is causing problems, which could become a major problem in the world. I mean, HIV infection was a good example of that. So I think that because Africa is the source of so many serious viral infections, and there are also other ones, obviously Mpox and Ebola and other things as well, which are often come from Africa. Those, the awareness and monitoring of those pathogens when they arise is very, very important. It's important that Africa is on top of that, and if it is on top of it, then it has a huge amount to offer the rest of the world, because there's the old adage, nobody is safe until everybody is safe. So it doesn't matter if the pathogen comes out of Africa, it could very well spread to North America and Europe and wherever else. So it's important that Africa be on top of this, and it can contribute significantly by making people aware of what the new pathogens are, their significance, and what problems they can cause.
Yes, the other part of the question, which is your connection to the cancer community.
Yes.
Yeah.
Well, I'm interested in cancer because of the relationship between hepatitis B virus and liver cancer. And as I said before, liver cancer is a very, very serious cancer, and it's important that it be prevented. The cancer can be prevented by preventing infection with hepatitis B virus, essentially, because the two go hand in hand. If you look at a map of Africa and you see where the high prevalences of chronic hepatitis B virus infection, and you overlay that with the prevalence of, or the incidence of liver cancer, they basically match each other. So my interest really is in the relationship between liver cancer and hepatitis B virus, and obviously preventing through vaccination or treatment to limit the limit the cause of the cancer arising. I have been involved for many years with the Cancer Association of South Africa, which is an organization that funds research and raises awareness of cancers in Africa. And it does, also contribute to funding research to develop improved management of cancers and prevention of cancers. So I was involved for many years, but am no longer directly involved with the Cancer Association of South Africa, but they only indirectly am I involved with them. So they continue to do excellent work. So my involvement with the cancer community is less strong now than it was before, but that's just because I have got other tasks that I have to see to and cannot focus as much as I did before. But I mean, it's a vitally important thing to be aware of the cancer landscape in Africa and its relationship to infectious diseases and non-infectious diseases as well, non-infectious causes of cancer.
Beautifully described. So, any last words for me?
I think we've covered just about everything, Paul. Nothing springs to mind that we've left out.
Yes, I think that was really, really extensive. Yes, thank you so much for talking to me. I really, really appreciate your time.